Okay, now let's talk about the chromatographic basics. Okay, so what do you mean by chromatography and why we use chromatography? Normally, chromatography is a technique of separating things out, separating especially protein particles or in some cases non-protein particles. How we can use chromatography to separate them, and what are the principles behind this chromatography? Let us discuss this. So let's move on. Yeah. So what is chromatography? Chromatography is the ability to separate molecules using partitioning. So what do we mean by partitioning? Let me take a color for explaining. So let me take this. Yeah. So what do you mean by this partitioning region? Partitioning means, in this case, partitioning characteristics of molecule remain in a stationary phase versus a mobile phase. So what we are having here is a stationary phase, a mobile phase. So basically, chromatography is an interaction of two different phases and the molecule to be separated. So for example, we have a stationary phase. As the name suggests, the stationary phase molecules are stabilized or fixed in a particular region. On the other side, I have the mobile phase, or the molecules in this mobile phase always mobile. That means they can be transferred from one place to another place, they are not stationary. And we have the sample molecule. We need to separate. We call it analyte or A. So we have these three different types of molecules. So what chromatography is? Chromatography means simply the separation, or sorry, it's not separation, simply the interaction between this stationary phase and mobile phase along with this analyte molecule. Okay, so analyte have an interaction with the stationary phase and it has an interaction with the mobile phase too, and all of those interactions are summed up, we end up with chromatography. So chromatography is a principle which is using partitioning. So what is this partitioning actually? The partitioning is the interaction variations between the stationary and mobile phase for an analyte. So in this case, the analyte, or the thing to be analyzed, is called the analyte. So this is the sample molecule we need to separate. So it's a mixture. It's a mixture of several different kinds of molecules. Okay, we need to separate all those different molecules from this mixture. How we can do that? Using this chromatography partitioning. So this molecule can have an interaction to dissolve in this solid phase. It can also have an interaction with this mobile phase to dissolve itself in the mobile phase. Okay, so sometimes the interaction between this analyte with solid phase is more than the interaction with mobile phase. In those cases, these molecules have a tendency to stick with this stationary phase molecules. So as a result of it, they cannot pass the tunnel easily. Okay, in reverse cases, sometimes the analyte molecule have a tendency to attach with mobile phase. It has a higher dissolve condition with this mobile phase. So in those cases, it will flow rapidly rather than sticking with the stationary phase molecules. Okay, so this is the basic idea between chromatography. So the interaction, as I've told you, can be based on the dissolving power, or it can also be based on the polarity. In some cases, the analyte can be polar. In some cases, it can be hydrophobic. So according to the difference between these different analyte molecule, we can use different types of stationary and mobile phase. We need to choose this stationary and mobile phase little bit more carefully to have or to achieve a perfect separation from the analyte mixture. Okay, for example, and another important thing is that we need to choose the stationary and mobile phase molecule both having a same characteristic. That means if we have a polar stationary phase molecule, then we must have a hydrophobic mobile phase molecule. Why is this? Because if we have both molecules hydrophobic in nature, then the stationary phase molecule can interact with the mobile phase molecule and whole system will be hampered. So you don't want that. So now in a nutshell, if we talk about this system, what is that? Is simply a tunnel. So think about a tunnel, and in this tunnel what we have a stationary phase. So let me change color for the stationary phase. So let's pick black to denote the stationary phase. So you have the stationary phase here and here in both these places, and let's change the color to green for the mobile phase. So we have the mobile phase. So the mobile phase is going in this direction, and we have stationary phase in both the sides. Okay, so if we have molecules which are hydrophobic in nature in the stationary phase, and we can if we also have the hydrophobic mobile phase, which is here, if both the stationary and mobile phase is hydrophobic in nature, then they can interact with each other. Remember, the hydrophobic regions interact with each other to form hydrophobic interaction, hydrophobic core. As a result of it, the whole system will be damaged. Actually, the chromatographic technique is based on the differentiation using the stationary and mobile phase molecules. So we need to have different characteristics of the mobile and stationary phase, that they should not interact with each other. Okay, so for example, we choose this stationary phase hydrophobic, and we choose this mobile phase hydrophilic in nature. As a result of it, they do not interact with each other, and the mobile phase can flow really quickly throughout this stationary phase. Okay, and mobile phase flow helps these analyte molecules to pass from one place to another place. So what we need to have a chamber here through which we can add these analyte molecules, and we have a chamber. It is called the injection chamber. Through this injection chamber we add analyte molecules, and using the force created by the mobile phase molecules, we travel these analyte molecules from one direction to this another direction, okay, according to the flow. Okay, and throughout this system, now the ability of this analyte molecule to interact with stationary phase and the mobile phase is checked. If this analyte molecule have a tendency to attach with the stationary phase, then it elutes very slowly, and if the analyte molecule have a better interaction with the mobile phase, then it elutes really faster. So according to the basis of elution, we can determine, or we can have a curve, we can detect it. So to detect the system, we have to have a detector, and we have this detector. So this is the detector here. This detector detects the elution property of those mixture analytes. And not only we have a detector, but the detector is attached to a monitor screen, and you can look at to this monitor screen, and looking into this monitor screen, we can make a curve. We can have a curve like this. This curve is called a chromatogram, because this is the written portion or enlisted type of our recorded data. By looking at this chromatogram, we can say about the property as well as the amount of those analytes that are present in the mixture. Okay, so we can both quantify it and we can analyze qualitatively. Now let's move on. So now it is a chromatographic separation. For example, let's take it. So we have two samples, A and B. It is mixed together to have a mixed sample. Okay, and we also have here is the mobile phase. Mobile phase is nothing but for example, it's a hydrophilic, so it's a water, for example. Okay, and the mobile phase, the action of mobile phase is to enter into one point and is to elute from the another point. So this is the function of the mobile phase molecules, and mobile phase molecules have a force, and that force helps to take or to make the flow of the analyte molecules. And we add samples, and sample is traveled from one place to another place, in this case from this A place to the B place via these mobile phase molecules, which is water. So water carries all these things, and water is eluted rapidly, but it carries this sample A and B. In this picture what we are looking at, this sample A, which is in color yellow, have a higher tendency to attach with the stationary phase, which is colored brown in this case. Okay, so in this case the yellow stick to this brown stationary phase, and the mobile phase passes out. As a result of it, the B elutes first and then elution of A happens. So after having a curve like this, where we have first B and then A, we can tell which type of molecule we are dealing with. Okay, and a question can be arise from this point, then how we can say which type of molecule they are, because we don't know how, we don't know something, so we need to have something to compare with this curve, and we have a curve which is a standardized chromatogram. So we have the standard chromatogram, and in the standard, using the standard chromatogram, we compare the result with the standard chromatogram, and by doing this we can achieve a relative position, and from those relative position we can say that this molecule is that and that molecule is that. Okay, so what is actually the chromatogram? The chromatogram is actually, it is the first application is done by MS Sweet, in 1903. Okay, so why it is called a chromatogram? Because we always need a detector to identify that, and also it is something related to the chroma, which means color, right? So we need a detector to detect it. For example, we can say it, we have eyes and we have nose. We close our eyes, and if we close our eyes we can still tell which food we are eating, but with our opened eye we can tell exactly, or we can confirmly say that the right name of the food. So except for this eye, we can say which food I am eating, but we can guess a thing. So running of this chromatography is not the end. We need to observe the data, we need to feed it into the monitor screen, and finally we need to have a diagram. With the diagram, with the comparing of this diagram or chromatogram with the standardized chromatogram, we can tell which sample mixture we are dealing with. Now, in this chromatographic separation, what we have talked about mobile phase, we have talked about stationary phase. So what is a mobile phase? Which flows through column, which carries the analyte. For example, in case of gas chromatography, we have a gas as a mobile phase. That's why it's called the gas chromatography. In the liquid chromatography, we have liquid as a mobile phase, and that's why it's called the liquid chromatography. Okay, and in stationary phase conditions, stationary phase, we need to have some molecules which have a higher rigid structure for the stationary phase. For example, in case solid molecules and as well as a liquid molecule can act as stationary phase. Now the separation is based on the partition between the mobile and stationary phase. So this word partition, you need to have a clear understanding of this word partition. What do you mean by the partition? We have analyte in the middle, we have stationary phase, we have mobile phase, the interaction with stationary phase and the mobile phase. So this is the partition, the interaction between the analyte with the stationary phase and the mobile phase. So according to that, the difference between the interactions is simply called the partition. That's it. Now now let's talk about a chromatogram and what parts a chromatogram are consisted of. Okay, so in this picture what we are looking at here is here the injection time begins. So in this case the chromatogram starts with the point when the analyte is entered into the column. Okay, it is the injection time, or when we add our analytes. And after adding these analytes or injection time, after a few time we have a peak, and here another peak. So we can find this peak. So what does this peak suggest? This peak suggests that the elution of different molecules. Okay, the elution of constituent of a mixture sample which is to be analyzed. Okay, so first peak is observed after some time. We call it the Tm. Right? We call it the Tm. After few time we have another peak, it is observed, which is we call it the Tr. It is also can be called the Tm. What do you mean by the Tm? Tm means the retention. Sorry, Tm means it is a time at which an unretained analyte or mobile phase travel through the column. Okay, or the mobile phase, it is the time for a mobile phase to throughout the column. After some time we have a peak. These peaks are called T. The distance between these peaks are called TR. First of all, let's talk about it is little bit more detailed. So you have a graph here. We have a detector sensitivity in this part, in this y-axis, and we have time in this x-axis. Okay. So for example, the chromatography begins, the injection is done, then after some time we have a peak. Then after some time we have another peak, and so on. So what we are having in this case is the peak observed from the injection time. We call it the retention time, or the time between the injection and the detection of an analyte is called the retention time. So this is retention time, or TR. Okay, and we can also have a retention time TM, or this is the time at which an unretained analyte or a mobile phase travels through the column. Okay, so this simple part here is called the TM, and very first thing arises here, the time is TR, or the retention time. Okay, so mobile phase can travel really faster through the column, but after that time, after some time, we can have a peak. That means after some time a molecular mixture, a molecule from that molecular mixture can be eluted. Okay, so that's it. And we have another relative retention. This is another nomenclature here, which is also called the separation factor. So what is the separation factor? In this case it is denoted with alpha. It means that the TR2 by TR1, that means for the distance between the injection time and the injection time and the detection of the analyte 1, so TR1, and the detection and elution between analyte 2, which is TR2. Okay, so so if we divide that TR2 by TR1, then we will end up with alpha. Okay, so alpha means a separation factor. So how well a mixture can be separated, it is denoted with the separation factor, or alpha. So if we have alpha more than 1, then we have a good quality of separation, or a greater quality of separation. So the more higher the alpha is, the more or the greater the separation can be. Okay. Now let's talk about another factor. It is the capacity factor, or it's called the K. It is denoted with small k. So what is this capacity factor? It's simply the adjusted retention time divided by the TM. So what do you mean by adjusted retention time? Adjusted retention time is simply the division between the retention time and the time for the mobile phase to elute. So the time for the mobile phase to elute is the TM, or the time for the mobile phase to elute, and the retention time is denoted with TR. So the adjusted retention time is simply the difference between this TR and TM. Okay, and the capacity factor denotes this adjusted retention time divided by TM, or we can have another denotation in this case. If we use this K term, or the capacity factor term, in case of the separation factor, then you can write like this: alpha equals to K2 by K1. Okay, so this is a different types of math. If you don't understand or don't try to understand, that's fine. But the thing is, you need to learn the map, the basic concept. The concept is the map begins with a dot, with a point. The point is called the injection point, where we add the analyte which is to be observed, or which is to be analyzed. After some time, when the analyte is detected, then we can have a peak. So whenever we have a peak, that means at that point the analyte is detected. At that time the analyte is detected. For example, if we run a chromatogram, in this case, after 30 seconds it detects the first component here, and for example after 102 seconds it detects the second compound. Okay, so in this x-axis order we have the time, Okay, you have the time. Okay, so this denotes actual the time, and we have the detector response throughout the place like that. So detection over time. So this is what we are having here. So the more higher, the peak or the peak height tells us about the amount of the analyte molecule, and also the area suggests us the amount, but the peak suggests us the presence of a certain compound. So if we find only the peak in time, we can say the we can use it in qualitative analysis, but if we want to do the quantitative analysis, we need to look for the peak height, and more exclusively we need to look for the peak area, which is the area between this peak. Okay, okay, so the retention time is simply the measure of identity. It is the same for all compounds. You can look at this. So it is over time. So we have time in this x-axis, and it is in the y-axis here, the detection response, and you can find something like that. Now these are the some types of chromatography that we will gonna look at. So these are the basic some types of chromatography, and in this case we have the absorption chromatography. So the chromatography can be identified based on the different procedure, based on different ways to separate between or best way to partition between the mobile and the stationary phase. So we can partition it using the adsorption. So what do you mean by adsorption? In this case, of solute on the surface of the stationary phase. So normally it can be seen for non-ionic compounds. The adsorption can be looked at the non-ionic compound, because the ionic compounds do not go through the adsorption. We have the ion exchange in the second place. That means the attraction of ions, not only the same types of ion, the opposite types of ions. So the anode, anion and the cation. So anion attracts cation, cation attracts anion, like that. And we can also have solubility as a partitioning. So this is not right. We need to have a solubility. We can also have a size exclusion. We can separate molecules according to their sizes. We can call it the gel filtration chromatography. Okay, we can differentiate molecules according to their affinity towards something. For example, some specific reactions of antigen and antibody. Some antigen is specific for some antibodies are specific for some antigens. So we can use their property of the affinity of binding with each other to separate them up. So this is the first one. This is the adsorption chromatography. This is for the non-ionic compounds. Okay, so these are the solutes. So in the mobile phase we have the mobile phase, these are the solutes. In this mobile phase we have the analytes, and this is the stationary phase. And in this case the adsorption between the analyte and the stationary phase via the surface interaction, that's why we call it the adsorption, which means the surface interactions of non-ionic compounds can be established. As a result of it, it sticks, or this analyte molecule sticks to this stationary phase, and the mobile phase passes through really quickly. By doing this, we can separate between a large amount of non-ionic compounds. In the second phase, what we are doing the partitioning chromatography. In this case, it is based on the solubility. It is based on solubility. So what is this solubility? Here, some solute dissolves in this coat which is around the stationary phase, and some dissolve less in this mobile phase. So some molecules which dissolves greater in this stationary phase coating will stay there, so they elute last, and some which do not dissolve in this coating of stationary phase elutes faster. By making this partitioning using solubility, we can separate some molecules. Okay, and we need to talk about another thing by using this partitioning, which is called the partition coefficient, which is denoted with K sub D. So the K sub D equals Cs by Cm. So just forget about them. Just think about the concentration of the stationary phase divided by the concentration in mobile phase. So this is the basic principle of all chromatographic techniques based on the thermodynamic equilibrium. For a reaction, for this kind of chromatographic reaction to be occur spontaneously, we need to have the concentration of stationary phase by the concentration of the mobile phase greater than 1. Then only this thing can be possible. Okay, Now let's talk about the ion exchange chromatography. So what is the ion exchange chromatography? As the name suggests, the ion exchange chromatography is everything to do with the ionic compounds. So what we have, again, in this case we have mobile phase and we have analytes, and in the stationary phase we have this. So the analytes, which is dissolved in mobile phase, analytes are negatively charged, for example, so they are anions in this case. And in the stationary phase we have lots of positively charged residues are attached to those stationary phase. Okay. So stationary phase, we need to choose the stationary phase for the ion exchange chromatography, which have to have different ionic concentrations. In this case they contain the positively charged ions or the cations. So we have lots of cations in the surface, and we have analytes which are anions. So the anions are attracted towards the cations, and the mobile phase can run off really fast. So the anions or the analytes have a tendency to attach with these cationic portions. So they elute last, and the mobile phase elutes really faster. So as a result of all this thing, and it finally ends with the separation of anion molecules from the cation molecules. So if we have other molecules which are cations, they cannot be attracted by the stationary phase molecules, so they elute really faster. Okay, so by using it. But in all these cases, in this ion exchange chromatography, the separation, in all the separation purposes, remember these things very carefully. We have to choose the stationary phase molecule and the mobile phase molecule really carefully, because in this case we need to choose the stationary phase molecule attached to the positively charged residues. Unless this, we cannot separate the anion factors. So in this case it is called the anion exchange chromatography, or the stationary phase molecule is called the anion exchange resin, because they are designed or programmed to take on only the anionic molecules, not the cationic molecules. That's why they call the anion exchange chromatography. On the other hand, we can have the cation exchange chromatography. In those cases, the stationary phase molecules are designed to take only the cations. And here we have the molecular exclusion chromatography. So what is this molecular exclusion chromatography? You can also call it the gel filtration chromatography. So this is the filtration. What is a filter made up with? It made up with pores, like, so pores are there, small particles can pass through, big particles cannot. But in this case we have also have spores in the gel, so we can run it through the gel, so that those gels having spores, but the small molecules can travel through the spores, and when they enter to the spores, because they have the small size, they entrapped in those gel messy networks. On the other hand, those large molecules who do not have are the sufficient for entering into the gaps can be eluted really faster. So in these cases, the huge molecules comes or elutes first, then the smaller molecules. So what we have here, we can differentiate between the big molecule towards the small molecule, or we can say in this way, we have higher molecular weight towards the lower molecular weight molecules, right? So we can separate some molecules using the molecular weight. That's why it's called the molecular exclusion chromatography. Now another is there, it is called the affinity chromatography. We have talked about it. What is the affinity chromatography? In this case, the the stationary phase is there and the mobile phase is there, which is going towards this direction, and mobile phase contains some analytes. The analytes are some types of chemicals or some types of molecules. Those molecules can only be attracted or attached with the specific types of molecules that are present in the stationary phase. So again, we design the stationary phase having, for example, in this case, some antigens. We have some antigens present in the stationary phase. So those antigens can only bind to only a specific type of antibody. Okay, so in this mixture, whole mixture, we have the antibody. For example, let me change the color. We have antibody here, antibody number 1, antibody 3, antibody 4, antibody 2. So antibody number 1 and 2 have the specific regions to bind with this type of antigens which are present in this stationary phase molecule. So they bind with this stationary phase, but all the other antibody molecules cannot bind, they passes through it, so they elutes faster. So again we are using the partitioning capability between the stationary phase and the mobile phase. Using analyte at the middle, we can separate them. So this is the basic way of separation of all these things. Okay, now here we have a question for you. Take the black, yeah. So here we have the question: find the adjusted retention time and the capacity factor for the benzene and toluene assuming the methane is unretained. So what is the case of this unretained? We know that the time distance between the injection and the component which is unretained is called the Tm, or the time for the mobile phases. So this is the mobile phase, Tm. So it is 50 seconds here. So it is 50 seconds. So from this picture we can say Tm equals to 50 seconds. Okay. And then we have to find the adjusted retention time. Remember what is adjusted retention time? It is the distance between the time distance between the retention time minus Tm. So it is T prime sub r. This is the Tr minus Tm. So we have found the Tm. So what is the Tr for benzene? In this case, this is the Tr for benzene. So this is the Tr1, and again this is the Tr2 for what is that? Is toluene, right? So it is the Tr2 for toluene. So the Tr1, which is for benzene, here it is 250 seconds, and Tr2, which is for toluene, is 350 seconds. Okay. So what is the adjusted retention time? It is simply Tr minus Tm. So Tr in this case, for example, in both cases, in case of benzene the Tr is 250 minus 50, answer is 200 for benzene, and in case of toluene we have 350 minus Tm, which is 50, is 300. Okay, so 300 is for toluene. So these are the adjusted retention time for both benzene and toluene. Now what is the capacity factor? Remember, capacity factor means alpha. And what is this alpha? Alpha equals Tr2 by Tr1. So it's like that. Okay. Or we can also obtain this. Sorry. No, we have made a mistake. So not capacity factor is not the alpha. Capacity factor is, sorry, this is a mistake. So it is a separation factor which is alpha. Okay, but the capacity factor is simply K, or the capacity of the separation. Okay, so in this case we have K, which is Tr minus Tm by Tm. So in this case we have Tr for benzene, it will be 250 minus Tm, which is 50. The result is 200 divided by Tm, which is 50. 200 divided by 50 in this case equals to 4. So the capacity factor is 4. As we have suggested, if the capacity factor or K is greater than 1, then the separation is very good. So in this case we have a 4 capacity factor. That means the reaction is, or the separation, it takes place really good manner. Okay, it is a very effective separation. So in this case what we are looking at, the qualitative and quantitative analysis. So let's talk about a peak. The peak suggests, what a peak suggests? A peak suggests the detection of the analyte sample. Okay, so whenever we have a peak, that means the detection of analyte sample is observed, and the peak height and the peak area, which is denoted in this blue color. Okay, height or area is have everything to do with the quantitative analysis, but the retention time, which is Tm, or sorry, retention time, which is Tr, denotes the qualitative state for that analyte sample. Okay, so retention time, Tr, have everything to do with qualitative thing, and the peak area or height has everything to do with quantitative thing. Here we have the plumber's view of chromatography. If you look at the plumber's view of chromatography, again, chromatography means stationary phase on solid, which is solid, not have to be solid all the time, but that has to be stationary. So at one stationary phase, one mobile phase, and one analyte, and what they have to run through a column. Okay, in all these cases. So in this case, we have an assumption. In this case, the mobile phase will always be faster than the analyte. That's a very good assumption, according to the plumber's view, or according to plumber. Okay, why is that? Because mobile phase carries these analytes, but still mobile phase is always faster than analyte, because mobile phase have nothing to do with the stationary phase, because we choose mobile phase molecules in such a way that they cannot interact, or to minimize the interaction between the stationary phase. But the analyte molecules can have the interaction with the stationary phase greater than the interaction with the mobile phase. So again, we have the stationary and analyte interaction, we can have the mobile and analyte interaction, but the stationary and analyte interaction can be greater than that, or it can also be less than that. Okay, not it can be less than that. Okay, so both the scenario can be present according to this nature. That's why the analyte would be slow. So in the first case, where the analyte have a higher tendency to attach to the stationary phase, it always moves slowly. So this is no problem about this scenario. But in the second scenario, where the analyte have interaction with the mobile phase, why still it eludes slower than the mobile phase? Okay, because mobile phase carries analyte. Okay, so whatever the analyte is, it is dissolved in the mobile phase. Okay, so when it is dissolved in the mobile phase, that means that's why the velocity or the force needed for separation, in this case provided by the mobile phase, is always higher, so it eludes faster than this analyte. Okay, so this is the thing. Now let's talk about the resolution of a separation. Okay, so in this case, resolution of two peaks from one another is the resolution of the separation. So whenever we have a distinct peak to be isolated like this, these are the distinct peaks, so we can say that this separation between these two different analyte molecules are greater than the peak found here, and it is greater than that and greater than that, because this is the most bad. So the resolution is getting lower. That means we have the peaks we are finding are merging with each other, so we do not find any distinct peak. And in this case, when you find a distinct peak, that means that suggests that this resolution is good, or the separation is good. Okay, so if we have the resolution more than 1, the separation is good. If we have the resolution less than 1, the separation is not good at all. Okay, and this resolution of separation can be measured with a formula. We call it this, the del Tr by the average width. Okay, so in this case, del means the difference between, Tr means the retention time. That means we have Tr2 minus Tr1, so that can be del Tr. Okay, and what is the W in this case? It is the width. So width is this portion, which we can measure the width at the basement, we can measure the width at the top. So this is the width. In most cases, we usually measure the width at the half height. When the peak have the half height, we measure the width. So width is simply referred to the half height width for that peak. Okay, so we have the, so normally we can have the width here, we can have the width another here, another here, we can average that, so we call it the width of average. So width average, so we divide it with the width average. This is the resolution of a separation, and we need to have the resolution of a separation more than 1.5 to have a good separation between analyte molecules. Okay, now here are some mathematical problems. In the first problem is worked out, so let's move on to the second problem in this case. So what happens if the peak elutes at peaks elute at 10 second and 10 minute and 10.4 minute, and a widths of 16 and 20 seconds. Okay, so what we have in this case, we have the peak elutes, so we have a retention time. So we have TR1, we have TR2, and what we have is the width. So let's make a width average. How can we make the width average? Let's add 16 and 20 together, which is 36, divided by 2, that means it's 18. So this is the average width, which is W average. Okay, now in this case, if we minus this TR2 from TR1, that will end up with 0.4, which is minute. So if we need to convert this minute into second, we can do by multiply it with 60, because 1 minute equals 60 seconds. Okay, by dividing it with 60, we end up with 24. Okay, end up with 24. Okay, and so what we have here is the difference between TR2 and TR1, which is del TR, and we have the average width, which is WAV. So the formula for the resolution is del TR, sorry, so let me erase this. Yeah, so it's technical fault. Okay, so the formula is del TR by W of average. So in this case, del TR is 24 and the average width is 18, so the result will be 1.33, which is given here. Okay, so that's how the result come to be existed, right? So these are the simple mathematics that you can face in your exams. Okay, so the resolution means a better separation of different portions or different molecules of an analyte mixture. So analyte mixture is made up with different types of molecules. The more we can separate between different types of molecules, we can call it the more resolution between them. Okay, so just go on through this. Okay, now here, why do peaks broaden? In this case, what we are looking at is bandwidth is proportional to the diffusion coefficient. You have known the diffusion coefficient is KD is proportional with the bandwidth. So if we have the higher KD, we have the higher bandwidth, and KD is composed of CS by Cm. CS means the concentration at the stationary phase. So if the concentration of analyte molecule is higher in the stationary phase, that means if the analyte molecule have a tendency to attach with the stationary phase molecules, then the diffusion coefficient is getting higher, and when the diffusion coefficient is getting higher, we are having broad bands like this. So bandwidth is getting increased. So in this case, remember, the KD is increasing as well as it is increasing. So the more analyte molecule is to attach, or it choose to attach with the substrate stationary phase molecule, it will elute last, right? Because it have a tendency to attach with the stationary phase molecule, and the mobile phase is passing out, so it have a tendency to elute last. In those pictures, we can find the bands at the last position. That's why if we draw a curve like this, detector and time here, we can find the first bands which are really close is like this, like having a really thin peak like that, and but the end peaks are slightly broadening like that, because the more this end time, the time is getting by, so some these analyte molecules need to elute really last or the later times. That means those molecules must have a tendency to attach with the stationary phase. As a result of that, they attach to the stationary phase, so they elute last, and when they elute last, they have a higher KD because of the concentration at the stationary phase. So higher KD means broaden peak. So this is related. Now, how good is this column for separation? We can determine the measurement, or we can determine the specificity, not the specificity, the activity of a column, because all this reaction is taking place in columns. So we need to have a column where we have the stationary phase, and through which we can run the mobile phase along with the analyte molecule. Okay, so how good a column is, we can measure that. We can measure that using another formula. In this case, this is called the number of theoretical plates, or caps n. So the number of theoretical plates, which is caps n, equals to 5.55 into TR divided by W half whole square. So this is a big formula. I don't want you to memorize this thing, but what is the basic concept behind that? Because we need to know how much a column is good for a particular separation, because we have different partitioning techniques for different types of separations. In case of gas chromatography, we have different sets of column. In case of liquid chromatography, we have different sets of columns. So we need to have different sets of columns to actually use different variety of partitioning to separate protein molecules. That's why it is very important to learn what is the capability of a column. So we can determine using this number, this n, which is in this case number of theoretical plates. Okay. Okay, and we can also compare between the different column efficiencies using this number n, which is number of plates or theoretical plates, it is more called, and also we can determine h. What was h? H is the related parameter in plate height. Okay, so if we compare between two different columns, we can have the related parameter, which is 8, and it means the h equals length of a column divided by the number of theoretical plates, or n. So this is the formula of h equals L by n. Okay, for example, there is a question here. On a gas chromatographic column, which is L having the 30 meter length, compounds elute in 5 minutes with W half of 5 seconds. W half means the white at the height. Okay, and it elutes in 5 minutes. So what is the number of theoretical plates and what is the plate height? The number of theoretical plates means the n. N equals, as we know, it's 5.55 into TR, which is the retention time. In this case, the retention time is 5 minutes. 5 minutes means into 60, 300 seconds. And we have the W half, it is given 5 seconds. So we had 5 here divided by 5 whole square 2. So we end up with this. So this is the number of theoretical plates for this column. Okay, so now let's find out the plate height, which is h. So what is the plate height? It is equals to L divided by n. So we have the number n. So what is L in this case? L is the length of the column, as we have 30 meters. So let it be converted into millimeter. So 30 meter convert into millimeter, we into this with 1000. After doing this, we have this and divided by the number, end up with 1.5 mm or millimeter. So this is the plate height. Okay, On the other hand, we have on a liquid chromatographic column which is having the L of 25 centimeter. That means L means the height of the of the column, which is 25 centimeter. That means let us convert it into millimeter. So we have to convert it into millimeter. So 250 millimeter. The compound dilute in 5 minutes, which is the retention time of 5 minutes, means the 300 seconds into 60. Okay, with the W half of 5s. So what is the number of theoretical plates? It is really relatively easy. So if 300, so this is the same formula is occurring here and the same formula is there. So you can find these results. So what are the necessity of using these huge mathematical formulas? But nothing but to compare with other different kinds of separation machines. We have different separation tools, we have different separation chambers, different separation columns. Now if we need to make a comparison between different columns, we can use these formulas to derive different results. And having or deriving different results, we can say that this column is better than that column. And by doing this, we can establish a whole system or whole data. We can arrange a whole data. Okay. So it is the Van Deemter equation. We'll talk about this in later times.